Okay, can we get on? It's not quite such a huge lecture as the last one I did, because the thing about homeostasis is it's kind of the entirety of physiology, and to try and cover that in like 1 hour is obviously a bit, or 50 minutes is a bit difficult. Today we're talking about skeletal muscle, and the specific remit is to talk about skeletal muscle as an excitable tissue. That was my remit. So as I said, I'm apologising a few times about the microphone not working properly. I've also got a bit of a cold, so if I go a bit croaky, I'll try and come back and say to the mic anything that sounded like it was important. Do feel free to tweet me any questions. I hope you've got these slides uploaded already. Were they on Vital, the slides? Did you look and they weren't there? Okay. I don't have access to Vital, but I'll ask them to put it up again. I'm sorry about that. If there was some way I could share it to you with Dropbox or something like that, that would be fine by me. Okay, so here's the flow that we're going to have on this lecture. What's meant by excitability in the first instance? Something about different types of muscle, different types specifically of skeletal muscle. Something about the neuromuscular junction, and then excitation, contraction, coupling will be the final thing. Okay, so I've ticked that. I ticked that, but I haven't actually said it, have I? Oh, it's all going out of control here. Not a good start. Right. What's meant by excitability then? So, excitability can mean various different things, of course, in lay terms. Generally speaking, in a biological context or an animal physiological context, when we say excitability, we mean very specifically it can fire an action potential. It can fire an action potential. That's kind of what it means. So I actually wrote a paper at some point where I used the expression, it's an excitable tissue and it fires an action potential, and someone was pedantic enough to say to me, but excitable means it fires an action potential, so that's a redundant sentence or something like that. So that's all excitability means. But of course, in a more lay term, any muscle is excitable in the sense that you can make it fire, so you could call that excitation, contraction, coupling, but it's not really what we mean by an excitable tissue. So the first subcategorization of muscle I'm going to give you is the broadest possible. We kind of basically have smooth muscle and striated muscle. So smooth muscle is what you would find in your blood vessels, your uterus, if you've got one, and the guts are all smooth muscle. Your sphincter, which hopefully you've got, is smooth muscle. Let's not get into sphincters just now. It's always going to end inappropriately. And then striated muscle, the other fundamental type of muscle really is striated muscle. Now, there's a little bit of ambiguity about what striated muscle means. To me, really, striated muscle is any muscle that's got stripes in it, and I'll show you an example of what I mean by that. That's got clear, visible stripes in it. But you'll quite often find that people use the terms striated muscle, skeletal muscle, and voluntary muscle interchangeably. So typically, if someone asks you about striated muscle, they don't mean cardiac muscle, even though it's got some stripes in it. They mean skeletal or voluntary. So voluntary muscle is all those muscles we have control about in order to walk, to work your hands, to work your jaws, etc., etc. And voluntary in the most general terms, because of course some of those muscles would include diaphragmatic muscles, the muscles of the diaphragm, which are necessary for breathing, and you can't stop breathing even if you want to. I'm sure some of you will want to within the next 15 or 20 minutes. And standing up, for example. Now, in my lecture plan, I was going to be standing up, and it's all going to plan so far in that regard. Standing up is something you're not conscious of, but clearly you can lie down if you want to. So that's voluntary muscle. They're constantly firing away to keep you in balance. So that's the definition of voluntary. Almost exclusively, voluntary muscle is synonymous with skeletal muscle. So cardiac muscle over here, that's very, very similar to skeletal muscle in a lot of regards. First of all, both of these skeletal muscle and cardiac muscle are both excitable. They're truly excitable. They fire action potentials very similar, in a very similar way to a neuron. And that's what causes the contraction to take place. With a smooth muscle, it's rather different. They don't fire action potentials at all. So the easiest, safest category is excitable versus non-excitable. Okay, within there, I think I've got it all. Let's show you the next slide, which has got some pictures. Now, I rooted about for these, and I'm not sure they necessarily convey exactly the pattern that I wanted to show to you. So skeletal, you can see the stripes in it. Those stripes are the sarcomere, and I'll be getting onto what that is later, although you have some idea from school, I should think. But on the face of it, it looks quite similar in this production with the lights and all this. It looks quite similar to cardiac muscle. In fact, to actually look at them under a microscope, it's not that difficult to tell them apart. Cardiac muscles tend to be shorter and fatter. Skeletal muscles tend to be much longer. So in fact, seeing as we've got all the mod cons in here, you will typically find that isolated in a dish, cardiac muscles kind of look like that, whereas skeletal muscles typically look like that with their stripes. Smooth muscle, on the other hand, even in this picture, it doesn't look particularly different, but when you see it under a microscope in a good image, a smooth muscle cell typically looks like a traditional cell that a kid would draw if you asked them to draw a cell. So that's kind of typically how a smooth muscle cell looks, i.e. it kind of looks smooth. Oh, that can stay there. No, let's tidy the place up as we go. Okay, so but essentially the similarities between, so the actual contractility mechanisms are quite similar in smooth muscle, but the excitation is different, it's absent. So in skeletal muscle, the excitation-contraction coupling is remarkably similar. I mean, if your specialist subject was studying excitation-contraction coupling within a cardiac cell, you'd probably say, oh, it's very different from skeletal muscle. But really, broad brushstrokes, it's a very similar kind of idea that's going on. Right, okay, so that's the main different types of muscle. But it starts to get slightly more complex because we have different types of skeletal muscle, and the remainder of this lecture will be about skeletal muscle because I think that was what people were thinking of when they asked me to do it. So basically you have two types of skeletal muscle, and they are rather unimaginatively named type 1 and type 2. Now, type 1 are the kind of the boring ones. Really, they're relatively thin, they're relatively weak, they go on for hours and hours and hours. They use oxygen just like most cells use oxygen. They're very efficient, and they'd be the kind of muscle that you would find predominating in a marathon runner. Fast twitch is much more exciting. They're the really bulky, powerful muscles that work in short bursts of activity, and they do that anaerobically, so they can do it without oxygen to get that burst of activity that you would need if you were sprinting. And there are a lot of biochemical changes or differences, morphological and biochemical differences, which underlie those two fundamental properties. And you kind of need to remember that type 1 is the boring marathon-y running type 1, and type 2 is the more sprinty kind of one, because from that you can kind of work out most of the physiological differences that there are, because everything, obviously in this kind of nature of a relatively well-evolved species, well, all animals are typically the same. These elements of physiology have very much evolved to suit the exact function which is necessary. So morphologically, you can see that this type 1 muscle fiber here, which is intended for marathon runners for long periods of activity without a great deal of power, well, they're thinner. And the fatter ones are those ones which are intended for bursts of high intensity force. So you don't necessarily need to remember all those other things. You can kind of work them out. Which ones are going to have a lot of mitochondria? Well, the ones which use a lot of oxygen, they're going to have a lot of mitochondria. So that would be type 1. In fact, the fact that one of them looks striated and the other one doesn't look striated, that's just not a very good photograph. Sometimes it's difficult to resolve the striations. And indeed, in my own labs, I've had a student look down the microscope, and I've said, oh, so you can see the striations in that fiber, and they'll look down, and they don't see any striations. And I'm kind of looking in, and I can just see the striations. I can't really see anything else. But it takes a certain amount of tuning in to sort of see these things. So in fact, they're both striated, and that difference is an illusion. You also find that that the type 1 muscle tends to be darker meat. So if you're going to have a traditional turkey Christmas lunch, you can be saying to each other, who would like type 1 muscle and who would like type 2, rather than light and dark meat. If you want to impress your family, bore your family, that would be, sorry. So it's darker because it's got more blood vessels. It needs more blood vessels to supply the oxygen because it requires oxygen. And it's also darker because it's got myoglobin, because of its oxygen need, it needs to suck myoglobin. Myoglobin, if you don't know, myoglobin is like hemoglobin, as is indicated by the name, but it tends to be resident within muscle. So it sort of draws the oxygen out of the blood. Okay, so I haven't really said anything about the type 2A and type 2B. Again, if we're sort of thinking in terms of examining and sort of remembering these kind of things, 2B is further away from type 1 than 2A. 2A is somewhere in between type 1 and type 2. So type 2 kind of goes either way, so to speak, whereas type 2A kind of goes either way, whereas type 2B is the kind of classic one, which I'm really discussing when I say that, make those comments about it. So now, sadly, to spoil my joke, I don't think anybody here probably ever heard of Diana Ross, who was, well, not only that, but the sound doesn't run, so that really kills it, doesn't it? So anyway, so there's the lovely Diana Ross song, I Want Muscles, or I don't care if he's young or old, all I want is someone I can hang on to. I want muscles all over his body. So there's a famous Diana Ross song from 1982, which I have been known to attempt to sing, but I think I should probably give that a miss at the moment. Anyway, so I was minded to think, what type of muscle was she talking about? Was she talking about type 1, type 2A, or type 2B? So typically, this would be someone who's got muscles all over his body, predominantly type 1. She's not bothered about how old or young he is, but this chap is. And he probably would be great if you wanted someone to hoe your garden the whole afternoon without complaining too much about the effort of it. Type 2B might be something more like this. And, sorry, type 2A might be something more like this. And type 2B might be something more like this. So I'm not sure which type Diana Ross was referring to, but there's 3 different alternatives. This one would be great if you wanted to carry a piano up the stairs, but not much good for it. Well, shall we say, carry the piano halfway up the stairs, probably, but not much good for anything else, in my opinion. Okay, so type 1, the skinny ones, they're using glycolysis. They're using oxygen and all that biochemistry you'll be covering. They're using oxygen. They're putting acyl-CoA into the Krebs cycle, and they're producing masses of oxygen. So in fact, they're really, really efficient. And that's why it's good for marathon running. It's not the absence of weight and the absence of bulk which is good. It's that raw efficiency, that with a very small amount of oxygen, you can go a long way. And that's what makes it special. So, but to give you an indication of it, have I written the number up? Yeah, basically, from glucose, you can make about 36 36 ATPs. And of course, ATP, as I'm sure you lot will all know, is the sort of the currency, is the cliche that people use, the currency of energy in the body. This type 2 anaerobic muscle, so it can give you a burst of power in the absence of oxygen, but it still needs energy to do that. And it gets that basically from NADH, and it can do it on the plus side. It can do it in the absence of oxygen. In the bad side, it's an extremely inefficient. I think you're talking 2 or 3 molecules of ATP coming from that process. And of course, you also have the payback, and that is after having carried the piano for halfway up the stairs and unsuccessfully dropped it, come back down to the bottom, you're going to need to have some R&R for quite a while, some rest and relaxation for quite a while. with the aching, stinging muscles as the lactic acid is reprocessed as oxygen becomes available back to pyruvate, which can then be put into Krebs cycle. But the fact that there's very few mitochondria in these muscles is quite interesting. And that follows. You can actually change the types of muscle experimentally. If you stimulate muscles in the right way, you can get them to behave like type 1 and type 2. I say that probably sounds a bit random to you, but I only discovered that a few years ago when we were putting a grant application on such things together. And I thought that was interesting. So from my experience, students love to learn stuff. So here's a list of facts, which are basically saying, giving you itemized list of everything said. I'll just have a look through and make sure I've kind of covered everything. So cell diameter, yeah, conduction velocity. So I was reticent to put slow because they all conduct pretty fast. They all conduct pretty fast compared with, say, a sensory neuron, which might be like half a meter per second or something like that. They all I don't know what the speed is, but they all conduct fairly fast. But type 2 do conduct faster. The actual degree of excitability, you need a bigger pulse of electricity, which we'll be getting onto, in order to make the type 2Bs fire. I think everything else is self-explanatory. Force, weak, strong, oxidative, speed, stamina, mitochondria. Yes, I've covered all that lot, so that's there for you to look on your slides, which let's not go into that anymore. Right, okay, so neuromuscular junction. Well, how does the electricity from, which that's what it is, coming from a motor neuron, so you send a signal from your brain down your spinal cord in your motor neurons, your upper motor neurons, they synapse and then they go out to the muscle to cause it contract. And that's the way they all work. That's the way they all work with a voluntary response. How does that action potential get from the nerve into the muscle? Well, the skeletal muscle fibers fire action potentials very, very similar action potentials to nerves. So they have an action potential very similar to nerves. So that means they sit at rest at whatever. I mean, minus 60 is a typical kind of value. Quite often people quote values of like minus 90 millivolts or minus 97 millivolts or something like that. But that tends to be from frogs. In humans and most mammals, it tends to be much more depolarized than that, more like minus 60 or minus 70. But anyway, it sits there at the resting membrane potential. And if you give it a small pulse of electricity, which will come from the neuromuscular junction, you get a small depolarization. If that depolarization hits the so-called threshold potential, then you have positive feedback. It all fires. You get sodium rushing in, and you get a depolarization. And then, of course, you get the repolarization with time as the sodium channels inactivate and potassium channels start to kick in. And it overshoots a little bit because there's a lot of potassium channels left open. And then it comes back to rest. So the most obvious difference between this and a typical nerve action potential would be that it's longer of duration. So 4 milliseconds. So they're still pretty fast. As I said, I'm reticent to call them slow. They're still pretty fast, but not as fast as a neuron. Considerably faster than a cardiac action potential, which I'm sort of not talking about, but they are interesting also, which is long and has a long slope like this. And because it's long and has a long slope, with lots of ion channel activity in that, it's the sort of thing that's fairly easy to modulate if you want to make a heartbeat faster or half slower, because it has this sort of long, and a cardiac muscle fiber, because it has this sort of long plateau, it gives you the option to use drugs which will shorten it or extend that period. And in fact, also, dangerously, sometimes those potassium channels get blocked by drugs as off-target effects and can cause sudden death. With a skeletal muscle fiber, it's more robust, it's faster, it doesn't seem to fail. It doesn't really seem to fail. There are various, of course, skeletal muscle diseases which are stuck in the, resident to the skeletal muscle, but they very rarely seem to involve anything wrong with the action potential itself compared with, say, a cardiac muscle cell. Right, okay, so as I said, the main difference is longer. I tried to draw this a bit longer, but that hasn't sort of come out, and I started trying to fiddle around, make it longer, and the figure started going manky, so I gave up with it. So basically, you have an action potential, which is a nerve, and somehow that gets to the skeletal muscle. So I've put some detail in there, which is stupidly beyond what you need, but I suppose it's a useful reference for me in that where people tend to think about electrophysiology as there being a voltage-gated sodium channel and a voltage-gated potassium channel. In fact, there are many different types of voltage-gated sodium channels and many different types of voltage-gated potassium channel. I think there are about 100 different types of voltage-gated potassium channel, and these are the ones which are found in skeletal muscle as opposed to nerves. KV 1.4 is a classic sodium channel which is involved with that. They have a lot of so-called KETP channels in it. I don't know if you've heard those before. They're a potassium channel, which open when ATP falls, and they can cause sort of fatigue of the muscle. And BK channels, which cause an after hyperpolarization. Don't forget, you also have chloride channels, which are important, and there are some diseases, some myotonia, which involve chloride channels as well. So basically quite similar but subtle molecular differences in their action potentials. So how does the neuromuscular junction work? Well, you have a presynaptic terminal. It's exactly analogous to a synapse elsewhere. You have a presynaptic terminal and you have a postsynaptic terminal. The postsynaptic terminal has got a unique name for a skeletal muscle neuromuscular junction. It's called the end plate. The gap in between has got the same name, the synaptic cleft. And you have ACH released in packets going across and acting on nicotinic receptors. It's always nicotinic receptors. You have this enzyme acetylcholinesterase, which is quite important, and that's crucial to the operating of the system, because what's essential for the neuromuscular junction to function correctly is that it releases a pulse of ACH, and then the ACH goes away. If the ACH stayed hanging around, you would get a sort of a paralysis. Well, you would get a paralysis. And so, in fact, this is the target. Blocking this enzyme is the target for some chemical weapons. That's nice. Let's get off chemical weapons, shall we? Back onto slightly more detail. So I just thought I'd give you a second slide on that. That was a little dumbed down. Second slide on that. Which shows a couple more details. Nicotinic receptors, yeah, they are subtly different to the nicotinic receptors you would find in the brain. Yeah, subtly different to the nicotinic receptors you would find in the brain. And you might, if I haven't said this to you at this thing to you at some point before, you might wonder why, seeing as there are nicotinic receptors in the brain, which are what nicotine binds to, and there are nicotinic receptors in the neuromuscular junction, why is it that when you smoke it doesn't make your muscles paralyze your muscles or cause you to twitch? And it's actually because they are less sensitive to nicotine than those receptors in the brain by being subtly molecularly different, but it's still nicotinic receptors. And of course, if you had the same nicotinic receptors in your brain as you have in your muscles, then smoking wouldn't be much point. And if it was the other way around, smoking would kill you much more effectively than it kills you with the current setup. It would be more sort of immediate, though, wouldn't it? Okay, so how do you get the ACH out? Well, it's stored in vesicles. You release one vesicle at a time. Well, I say you release one vesicle. The ACH is only released in little packets, but of course you release many, many vesicles at the same time. Is that me? No, it's somebody else. Good. It's usually me when that happens. And what triggers it? What triggers it is the action potential comes down and it activates calcium channels, which let in calcium. The calcium is then a trigger for the fusion of those vesicles with the presynaptic terminal and the release of the ACH. So calcium is the trigger for that. Okay, I think I've got everything else on there. And so, again, analogous to a synapse, what happens is that the ACH, when it binds to the nicotinic receptor, it opens, basically, this depicts it as a sodium channel. That's a typical misrepresentation because, of course, in reality, the nicotinic receptor is a sodium-potassium non-specific channel. But opening that still depolarizes the muscle. And it depolarizes the muscle a little bit to give you an extra or excitatory postsynaptic potential, EPSP. And if that is of sufficient size, then it causes the action potential to fire. So a few small packets, a few small releases of acetylcholine don't do much. If you hit it with enough of it, you get your action potential to fire. Okay, so and then obviously the action potential travels along the skeletal muscle fiber and causes a tension to develop. Now that tension is almost instantaneous with the action potential, but it stands to reason that if you draw it correctly, you have your action potential just slightly preceding that wave of tension. But how does it do that? So this brings us on to excitation, contraction, coupling. So the basic structure of the system is that within a muscle-rich zone, like an arm, within a muscle, you have got a number of muscle fibers. So the lay term is a muscle, and within that, there's muscle fibers. Sometimes people call them cells, but they've got lots of nuclei, so fibers is probably more appropriate than cells. I'm personally not fussed whether people think of them as cells or not. They're kind of cells to me. So you've got a muscle, you've got a number of muscle fibers, which are equivalent to cells within them. Within each of those cells or muscle fibers, you have myofibrils, which are the fundamental tension-producing structures. So you have those bunched together within your muscle cells or within your muscle fibers, which are bunched together within your muscle, within your macro muscle. Okay, so each of these has got a distinct structure, which I'll be coming back into, which boils to, this is under electron microscope and then just a cartoon of it, where you have these two very important proteins for contraction. You have myosin, which is the thick one, and you have actin, which is the thin one. So somehow you have to get your nerve electricity and your neuromuscular junction all the way in, all the way down to cause a release of calcium here, because it's a release of calcium here in the sarcomere which triggers the excitation. And so what you have within the muscle is you have a T-tubular network, a so-called T-tubule network. This network is like, it's kind of like a bunch of nerves that run within the muscle cell itself, but they're not nerves, they're T-tubular. It's a T-tubular network. So the alternative, you just think of them as holes that run through. But they conduct electricity within. So what we've got here, again, I'm going to cover the I bands, A bands, and all that a little bit more, I think, in a moment. But what you have is wrapping right in around the, running right through the center of the muscle fibers, you have this T-tubule. And that's shown in purple for the non-colorblind. And then that comes very close to the sarcoplasmic reticulum, which is a network which then beds all the way through. So you kind of have these kind of networks of interconnecting tubes within the muscle, which allow the spread of electricity and electrical excitation right in to the contractile apparatus. So electricity flows in through the triad, and that signals to the sarcoplasmic reticulum to release calcium. Here it is in another photo. So this is the surface of the muscle. The action potential runs along the surface of the muscle. It bombs down the T-tubule, and it connects in a slightly mysterious way. It's still not known. I was absolutely intrigued by how this was done as an undergraduate, and I'm afraid it's still not fully resolved. There's a lot of data, but not any really specific way. Somehow the T-tubule connects to the sarcoplasmic reticulum and causes it to release calcium. It's important to remember that the area where the T-tubule and the sarcoplasmic reticulum and all that come together is called a triad. The release of calcium also can cause the release of further calcium, and that happens again in the heart, and it's called calcium-induced calcium release. Calcium-induced calcium release. But it's more profound in the heart, probably, than calcium, than in the muscle. Okay, so what does that do? So the calcium then binds to the tropomyosin. So you've got actin, myosin, tropomyosin, and troponin, and causes the binding sites for myosin itself to be revealed. So let's spell that out a little bit more. You have tropomyosin wrapped, which is not the same as myosin, wrapped around the actin. The actin itself has got binding sites for myosin. So myosin, big fat, has these feet that bind on to the actin and pull it along. That's how contraction works. But the little binding points for myosin on the actin are covered up by tropomyosin. So that in turn has troponin on, which has got binding sites for calcium. So the calcium really does act a trigger like on a gun. Well, it's not quite like a gun, but it's a trigger. It really is very much a trigger. It binds to the tropomyosin, causes a conformational change, which causes the whole thing to just shift over and reveal these myosin binding sites, which means that these pores, or these feet of the myosin, can then just grab a hold, latch a hold. And that's the trigger step that causes the contraction to take place. Now, how does the contraction actually take place? Well, it's the sliding cross bridges. Sometimes it's called the sliding cross bridges theory, but I don't think it's really a theory anymore. And in fact, one of the most fun experiments that I ever saw anyone do would be, I think they got a Nobel Prize for it, but sadly I can't remember what their name is they synthesized myosin in the form of little balls. Normally myosin is a hooky-like thing that goes along. And they formed myosin in little balls, and they got sheets of actin, and they put the myosin balls at one end of the actin, and then dripped on pulses of calcium in the presence of ATP, and they got the little balls to roll all the way along the actin. I thought that was really cool. I'm not sure it was terribly useful, but I thought it was quite cool. And it did help to explain and pretty much confirm the sliding bridge theory. So the sliding bridge theory means that whereas you would think that to generate tension you would normally pull on something, you don't actually pull on it. What you have is you have the actin and the myosin, and they slide against each other. Individually, the myosin pulling itself along like a little hook, pulling itself along, and of course individually a negligible amount of force, but when you have thousands of these things doing it together, they generate a significant force. And that's all taking place at the overlapping period, the overlapping bit of a muscle. So you can see that the more overlapping there is until it's at full contraction, the stronger it will be. So quite often you can get the maximum force if a muscle is part way contracted. When it very first starts, these bridges are sort of separated, only a tiny bit of overlap. There physically isn't much overlap to cause tension. As they overlap more and more, they've got the ability to generate more tension. And again, that's important. We're basically talking about skeletal muscle, but just for your information, that's also partly how the heart works. You put a big pulse into the heart and stretch the heart out too much. Stretch the heart out, you cause the overlapping bridges to overlap a bit more, and it gives more tension. So that's important. Right now, this sarcomere shows your H bands, your I bands, your Z bands and stuff, which is all terribly sort of confusing. The M in the middle through the center, which is unlabeled in this. So the Z band is very distinctive because that marks the attachment point for the actin. It would be handy if the A band stood for actin, but it doesn't. The A band delimits the myosin bit, which is a bit difficult. It's one of those things where, I remember, it's the A stands for actin, except it doesn't. So it's kind of like it ought to be called an M band, but it's called an A band, and that's where the myosin lies. The H band is sort of the gap in the middle where you only have the myosin, you don't have actin. And the I band is kind of the intermediary bit between the sarcomere where there isn't any myosin or anything much going on there. So they're the different structures, but you can see the myosin with its little feet, and they grab hold of the actin, and that's how the tension's delivered. Right, now, okay, so I probably should have put that one up slightly earlier. So obviously you can see, I think it's self-evident from that when it's contracted it goes like that, and it contracts no more, but it can hold quite a bit of force in that state because there's a lot of overlap going on. There we go. Right, okay, so I'll just take you through a cycle of excitation, contraction, coupling. Now this is taking place once the calcium has spiked and revealed the binding sites, because if the binding sites are blocked, there's no calcium and the tropomyosin is covering the actin binding sites, you don't get any movement. But the other thing you'll know is the so-called stiff. When somebody is dead and then or an animal is dead and they go through this phase of rigor mortis, that's as the ATP has gone away. because in the absence of ATP, you can't get any of this cycling. So in order to have cycling, you need two things. You need the ATP at an appropriate level to allow this process to take place I'm about to describe, and you need to trigger it with calcium. Either of those things on their own won't work. But in the absence of calcium, sorry, in the absence of ATP, as a muscle just sits there, it sits there with the myosin firmly bound to the actin. All the other binding sites will be blocked if there's no calcium. If there's calcium there, they're unblocked, but there's no ATP. It will still just sit there, locked against it. And why does it soften up afterwards? Because things break down, given enough time. Okay, so this is your resting position. This is your resting position. ATP is around, but it hasn't. But the calcium trigger has not come to free up any binding sites. Then we give calcium, we free up those binding sites, and ATP is present. And this is what happens. It's all in equilibrium, so the myosin dissociates from its current binding site and binds ATP. That causes binds ATP, that causes all that happens together. The ATP dissociates from the myosin dissociates from the actin. Okay, so then you get the ATP is cleaved into ADP, causing a conformational change, and the little myosin head reaches forward for the next available binding site. If there's no calcium, there is no binding site, but I've just said we've triggered it with calcium, so the binding sites have been revealed. So the myosin binding site reaches forward and grabs hold of the myosin head reaches forward and grabs hold of the actin. And then as the ATP is released, it causes a power stroke and another conformational change, and this head bends back to its starting position, which has advanced the whole thing forward one binding site. As I said, if you do it with a finger, it's no ATP, ATP binds, ATP converted to ADP, that comes off and it goes like that. So it really ought to be an animation, shouldn't it? And there you go, that's the power stroke, and we are back to the starting stage. That's how excitation contraction works. Okay, so as I said, that's not a massive lecture. I better just check. I normally have another slide I've forgotten about. I don't. If you've got any questions, please tweet me. If you don't get a version of these slides uploaded, both in stream capture and in the slides which have been available for a long time, please let me know and I will get it sorted.